Very good afternoon students. So now we are going to exercise the rest 20 questions of APEAPCET 2025 question paper. So now in this question, first question if you consider, this is from mineral nutrition. As how we have exercised previous objectives, same way we are going to decode each and every objective, looking into from which chapter that question has arisen and what are the respective possibilities of options to this. We will look into each and every concept of the chapter. Okay, concept wise objective practice will happen here. Fine. So now if you look into this question, this is from the chapter mineral nutrition. First option if you see, that is ATP synthase. He has given the sequence of essential elements potassium, magnesium, molybdenum, phosphorus and carbon of the given list which show the following activities he is asking. ATP synthesis is promoted by which molecule from this phosphate group? We all know ADP plus inorganic phosphate will give rise to what? For ATP synthesis. So, the phosphorus molecule is contributing in the formation or the synthesis of ATP. Second, component of enzymes. Okay, component of enzymes, molybdenum is one of the component of enzyme. Okay. Enzyme activation. Enzyme activation, most of the enzymes are activated by magnesium like DNA polymerase, RNA polymerase, topoisomerases, those are being activated by the magnesium. And turgidity maintenance, turgidity in stomatal cell if you consider, stomatal opening and closing is modulated by guard cells. In guard cells, based on the turgidity, opening and closing of the stomata will happen, and that turgidity is maintained by potassium ion concentration inside the guard cells. So, here what options we have to choose ATP synthesis is related to phosphate, component of enzymes that is related with what element? Molybdenum, enzyme activation by magnesium, turgidity maintenance by potassium. Clear, right? Next. Here match the following type of question. This is based on the diseases. Okay, diseases of the plant. Each disease is given and the plant is given with respect to this. Mottled leaf is observed in the citrus plant, especially for the leaf areas will get affected. Next, heart rot which occurs at the central region of the plant part, it is mainly seen in the beets. Mouse ear is observed in pecan and bronzing is observed in the Fabaceae members, those are legumes. Okay, this is direct question from academy. Okay, diseases and the plants, and this is from the enzymes of biomolecules. Okay, beta. So, from here he has from 6 classes of the enzymes, 4 are given here, enzymes, and the respective enzyme is given over here. Based on the function we have to match them. So, now if you see hydrolases, okay, hydrolases means what? They will cleave the water group. Okay, hydrolases, they will cleave the water group. So, here hydrolases belongs to the enzyme activity, that is fructose 1,6 bisphosphate, and ligase if you observe, that is with respect to glutamine synthase. Lyases is arginosuccinase. Yes, lyases is arginosuccinase and transferase belongs to hexokinase. Now, I will be telling you each and every enzymatic activity and why these enzymes are having this as examples. First is hydrolases, where fructose 1,6 bisphosphate is the example, right? Hydrolases, fructose 1,6 bisphosphate, wherein fructose 1,6 bisphosphate is converting fructose 1,6 bisphosphate to fructose 6-phosphate. This is removing the phosphate group. That is why placed under hydrolases. Next, lyases example will be arginosuccinate, which leads to the formation of arginosuccinate. Ligase, glutamine synthase, where you will observe this enzyme during nitrogen fixation. Next, transferase enzyme, which will transfer the phosphate group. Transferase, best example is hexokinase, which will transfer the from glucose, it will transfer to glucose 6-phosphate. Phosphate group is added to glucose by hexokinase. Transfer of groups is seen with respect to the enzyme transferases. So, here the options will go like hydrolases is for fructose 1,6 bisphosphate, lyases is arginosuccinate, ligase glutamine synthase, transferase is hexokinase. Clear? Next question. This is from plant physiology. He is asking us to identify the correct statement. This is based on the discoveries only, but statement based he has asked. Very easy to tackle, ma. Look into it. Joseph Priestley in the year 1770 showed that plants will have the ability to take carbon dioxide from the atmosphere and they will release out the oxygen. By what experiment? He said that bell jar experiment. So, yes, statement 1 is true only. Second, Jan Ingenhousz release, he said like release of carbon dioxide by plants was possible only in the night. Wrong. Jan Ingenhousz has conducted hydrilla experiment where he clearly stated that sunlight and chlorophyll are essential for photosynthesis and oxygen is liberated out of photosynthesis. So, this is wrong. Third, Julius von Sachs, he said that glucose is formed as the end product of photosynthesis. Green parts in plants produce glucose which is stored as starch. Okay. Then C.B. Van Niel. So, this is true only. C.B. Van Niel, photosynthesis is essentially a light dependent reaction in which hydrogen from an oxidized compound reduces carbon dioxide to form carbohydrates. That is true. It is C.B. Van Niel who said that carbon dioxide is reduced to glucose and water molecule is oxidized and oxygen is liberated out as byproduct from photosynthesis. That is said by whom? C.B. Van Niel only. Okay. So, now if you see all these three statements are correct except the second statement. Okay. So, 1, 3 and 4 are correct. Next. So, this question, number of ATP and NADPH required to make one triose through Kelvin cycle. Okay. In Kelvin cycle, we all know to fix one carbon dioxide, three major steps happen, that is carboxylation and then reduction. Okay. And then what? Reduction. During that, after carboxylation, it is fixed in the form of 3-phosphoglyceraldehyde or phosphoglyceric acid. This is reduced to glyceraldehyde 3-phosphate. Okay, this is reduced to what? Glyceraldehyde 3-phosphate, wherein for one carbon dioxide molecule 2 ATP and 2 NADPH are released. 2 ATP and 2 NADPH. And glyceraldehyde 3-phosphate will be regenerated back to RUBP. Here one more ATP is being utilized. As such, for one glucose molecule 6 Kelvin cycles takes place, 6 carbon dioxides are fixed and the amount of energy we need to calculate for 6 cycles. But here he is asking to make one triose through Kelvin cycle. He is not asking us hexose. Okay, he is not asking us hexose, he is asking us triose. For triose formation, 3 Kelvin cycles have to happen, right? So, 3 into 3 will be, for one cycle 2 plus 1, 3 ATP are used. So, for 3 cycles are required to form triose, right? So, 3 into 3 it is 9 ATP and 3 into 2. 2 NADPH for one cycle, for 3 cycles it will be 6 NADPH. Okay, it will be 6 NADPH. So, the answer will be 9 ATP and 6 NADPH. It is as simple as it is. Next. So, here choose correct statements among the following. Fatty acids are broken down to acetyl-CoA before entry into the respiratory pathway. True. During the time of aerobic respiration, after glycolysis, the immediate step that is happening is link reaction, where in link reaction acetyl-CoA is formed. Okay, acetyl-CoA is formed, and this acetyl-CoA is sent for tricarboxylic acid cycle. So this is true, correct statement. The F1 component of ATPase is integral membrane protein complex. Wrong. Okay. In integral membrane protein, actually ATP synthase will have F0, F1 channels. Okay, F0, F1 channels, where F0 will be embedded inside the membrane and F1 will be towards the matrix. Okay, as we are talking about mitochondria, F0 will be in inner mitochondrial membrane and F1 will be in the matrix. Okay, so this is not, F1 is not inside the membrane, this is wrong. Devansh, yes. So for what question you are answering, ma, type the question number and you then answer, beta. Yes. So now Hans Krebs elucidated the tricarboxylic acid cycle. Yes, that is why TCA cycle is also called as Krebs cycle. All living organisms retain the enzymatic machinery to partially oxidize glucose. True, because both in the prokaryotic, both in the anaerobic or aerobic organisms, okay, all the organisms wherever respiration is happening, glucose is the common pathway. In glucose, what happens? The glucose is partially oxidized to pyruvic acid. Okay, glucose is partially oxidized to pyruvic acid. Glycolysis takes place in all the organisms. So yes, this is also true. So what will be the correct statements? 1, 3 and 4. Clear, ma? Next. Question number 27, this is, look into the question properly. Plants follow different pathways in response to the environmental or phases of life to form different kinds of structure. Okay, this is to form different kinds of structures. So here if you see, plants responding to different pathways, either to phases of growth or to environment. Okay, plants will follow different developmental pathways either with respect to phases of growth or with respect to environmental factors. If it is with respect to phases of growth, okay, at juvenile phase one leaf structure is seen, at mature phase another leaf structure is seen. Best example for this will be coriander and cotton. Based on the environmental factors, best example is larkspur, wherein larkspur will grow differently in the aquatic habitat and it will grow differently in the terrestrial habitat. So this pattern of following different developmental pathways is defined as plasticity. Okay, this is known as plasticity, and the different growth patterns of the leaves is termed as heterophily. Heterophily is one of the branch under plasticity. Clear, ma? Next. Pleomorphic bacteria. Amongst the bacteria that are given here, Vibrioids, Spirochetes, Bigetova and Acetobacter. Acetobacter will form multiple shapes. It will vary in its shape based on the environmental condition. So, pleomorphic bacteria, Acetobacter comes under pleomorphic bacteria. Next. So, this you see, ma, complementary receptor site. Complementary receptor site is with respect to tail fibers, because tail fibers which have lysozyme, they are receptive to the cell wall of bacteria. Complementary receptor site, that is tail fibers. Two identical molecules of RNA. Two identical molecules of RNA are seen in Retroviridae, whose genetic material is RNA. Next, viral enzyme is what? Lysozyme, which degrades the cell wall of bacteria. Proteinaceous infectious particle is prions. Prions are just having abnormally folded protein. They doesn't have genetic material. So now match it up where complementary receptor sites are present in the tail fibers of the virus bacteriophage. Next, two identical molecules of RNA are seen in Retroviridae. Viral enzyme is what? Lysozyme, which is present at the tail fibers. Proteinaceous infectious particles are prions. Clear ma? Next. Choose the correct statement among the following. Fine. This is from genetics, defining the terms homozygous and heterozygous. If you are clear with homozygous and heterozygous, then you can easily answer this question. Heterozygous parent produces many types of gametes. Okay. Heterozygous parents will produce many types of gametes. That is wrong. Heterozygous parent will produce only two types of gametes. Okay. Homozygous parent will produce similar type of gametes. That is true. Heterozygous parent distribute two alleles to gametes. No, parents will not distribute the gametes. Okay. Incomplete dominance, F1 hybrid resembles either of the parents. Wrong. In incomplete dominance, F1 hybrid will not show the resemblance with any of the parent. This we have seen with the example of snapdragon, wherein when the cross of red colored flowers with white color flowers. Okay. Pure red color flowers when crossed with white color flowers, the F1 offsprings are of pink color, heterozygous pink color. This is not resembling any of the parent. So, yes, this is wrong. The correct statement is homozygous parents will produce similar type of gametes. Clear? Next, this you see ma, assertion reason question. This is from Human Genome Project and fingerprinting, DNA fingerprinting. Assertion, if you see, genetic maps are extensively used as starting points in the human genome sequencing project. That is true. Genetic maps are used as starting points for human genome project, because genetic maps will give you the complete genome which is present inside the organism. Reason why genetic maps are used: because frequency of recombination or crossing over between gene pairs on the same chromosome as the measure of distance between the genes. The more the distance, more possibility of recombination and crossing over, less the distance, less crossing over takes place. That is identified through gene maps only, right? Next, between genes and mapped their position is called as gene mapping. So, here in assertion they have mentioned gene maps are used in human genome project. In reason he has clearly explained why gene maps are used. So, with this, assertion and reason are true, and reason is correct explanation of assertion. This you see ma, completely this is from molecular bio, especially with respect to scientists and their discovery. The scientist identified the ratio between adenine and thymine and between guanine and cytosine are constant and equal. These are given from the principles of which scientist? Chargaff laws. Okay, so here the scientist who said that adenine and thymine and ratio between adenine, thymine and cytosine and guanine is constant, that is by Erwin Chargaff. Choose correct statement in the following. Okay. In Griffith's transforming experiment or in Griffith's transforming principle, the digestion with DNAs did inhibit the transformation, whereas proteases and RNAs did not affect the transformation. First, let us quickly recall what is Griffith's experiment. Griffith has gave the transforming principle. He said there is something that is transforming between the organisms. Okay. So, transforming principle is given by Griffith from the experiment of Streptococcus pneumoniae, and who re-identified the transforming principle, that is Oswald, MacLeod and MC Carte. Okay. Avery, MacLeod and MC Carte, they have said that DNAs, digestion with DNAs and DNAs did inhibit transformation, whereas proteases and RNAs did not affect the transformation. That is true. Next, virus grown in radioactive sulfur contained radioactive protein and radioactive DNA. In this experiment, okay, so look here, what happened is bacteriophage, okay, bacteriophage which is infecting virus. At the tails it has lysozyme, right? So this bacteriophage infecting the bacteria, radioactive isotope, that is sulfur, is placed at the protein coat, because protein has disulfide bridges, right? So sulfur is, the radioactive isotope of sulfur is placed with protein coat, and phosphorus is with genetic material. Okay. Now you look into the statement properly. After infection, blending and centrifugation, you observe the phosphorus, the radioactive phosphorus is not observed in the supernatant, because protein coats are placed above and the thick radioactive phosphorus is below, right? So in supernatant you do not observe phosphorus, radioactive phosphorus, only sulfur will be present, radioactive sulfur. So virus grown in radioactive sulfur contained radioactive protein and radioactive DNA. It does not contain radioactive DNA because sulfur is radioactive, it is present in the protein coat. Next you see virus grown in radioactive phosphorus contain radioactive protein and non-radioactive DNA. Okay. Virus that is grown in radioactive phosphorus contain, it does not contain radioactive protein, it contains radioactive DNA. Next, mice infected R strain die from pneumonia, but mice infected from S strain did not develop pneumonia. This is wrong. Mice which is with rough strain did not get affected, but mice with smooth strain got affected. Okay, so what will be the correct statement now? Option 1 will be true with respect to Griffith's principle, transforming principle. Now, this question is about restriction enzymes from biotechnology leads to formation of sticky ends in both DNA vector and foreign DNA. This is because of what? This is because of all the three options are relevant. This recognizes palindromic nucleotide sequence. Of course, restriction endonucleases will recognize palindromic nucleotide sequence. Cut of the strands of DNA a little away from the center of the palindromic sequence. Yes, cut is little away, and then this helps to form complementary cut counterparts. Okay, this will help to form complementary cut counterparts. So all these three statements are correct only. Next you observe bond formed by DNA ligase. DNA ligase will nick the two nucleotides, it will connect the two nucleotides. So the nucleotides are connected, one nucleotide at 3' hydroxyl group is connected to the other nucleotide at 5' phosphate group. Okay, other nucleotide at 5' phosphate group. This connection is, this bond is formed by what? DNA ligase. This is by phosphodiester bond. Clear ma? Next. This is from strategies only. Many a times the questions are asked with respect to Cry1AC, Cry2AB and Cry1AB. So here you see Cry2AB or Cry1AC. Just a second. Cry2AB or Cry1AC, they will resist, they will show resistance to cotton bollworms, and Cry1AB will show resistance to corn borers. RNA interference will prevent the nematode pathogen. How? Because RNA interference will mask or it will silence the target gene by sending a complementary RNA which is complementary to the nematode's RNA. Okay, nematode's mRNA, one more RNA is sent which is complementary RNA. this will silence the effect of targeted genes. Silence the effect of targeted genes. Clear? So yes, this is true. Next, Bt cotton. Bt cotton is resistance to pest. Clear? So this is the match accordingly. Okay, I hope the concept is clear with respect to this. Next, about transgenic plants. Okay, papaya is transgenic against ring spot virus. Bt cotton is resistant to insects. Tomato is resistant to Pseudomonas, and potato is against Phytophthora. Okay, this is directly asked question. So this is also from strategies ma. Wheat with Himagiri variety. Wheat has what variety? Variety name is Himgiri, not Pusa Komal. So this is wrong. This is against Hill Blunt, that is true. Cauliflower has two varieties, Pusa Subra and Pusa Snowball K1. This is against black rot, that is true. Chili, Pusa Sadabahar, that is true. This is against TMV, tobacco mosaic virus or chili mosaic virus or leaf curl. 3 are correct. Last is Brassica. Brassica variety is called as Pusa Gaurav. Okay, Brassica variety is what? Pusa Gaurav, which is against bacterial blight. So what are the correct statements, correct match here? What is the correct statement? 2 and 3, right beta? So the correct match is 2 and 3. Look into this question. This is from microbes in human welfare. Very simple question. Just by looking into this you can answer. Microorganisms that improves the nutritional quality of curd by increasing vitamin B12, that is cyanocobalamin. So what is that bacteria? Curd making bacteria, lactobacillus bacteria. That's it. It is very simple. If the question is with respect to cheese making, then you have to go for the option Propionibacterium shermani. If it is with respect to antibiotics, then Penicillium notatum. If it is with respect to alcohol making or brewing industries, then the option should be Saccharomyces cerevisiae. Clear, right ma? Now, the last question of today's session, just look into it. This is very simple again, common question only, just look into, yes. So, enrich nutrient quality of soil. Biofertilizers will enrich the nutrient quality of soil. Biopesticides will kill the insect pest, and biogas is decomposition of dung to produce gas, that is methane, okay, which is biogas. Bioreactor, growth of large number of microbes on industrial scale, that is bioreactor. Clear, right ma? So this is the complete session of the questionnaire today. From coming questionnaire sessions, we are going to discuss the top 1000 questions for NEET examination. Okay, completely all the set of questions we are going to look into. Match the following questions, statement based questions and assertion reason questions and diagram based questions. All these questions we are going to look into. Okay, so stay tuned and thank you.